So before our lecture start, I want to advise you one thing, that if you know all the basics of cell cycle, that what are the phases of cell cycle, what are the main events that are taking place in those phases, and what is the CDK, CKI, CDK inhibitor, NFS promoting complex, only then watch the video. And if you don't, then please try to understand them first. And if you do, please watch the video, enjoy it. Hello mobile learners, welcome back again with a new video, and topic for today's video is cell cycle regulation. Okay, and this is my first time that I'm making my videos in English, so I'm kind of feeling nervous now. And if you are somewhere outside from India, so please sorry for my accent, forget about it. So the topic is cell cycle regulation. Okay, so the concept that we are going to understand in this lecture is that, suppose these are the phases of cell cycle, like G1S, then G2, M. One more phase is Z0, it can reverse it, and M1 can, like this one. This is the cell cycle, the phases of cell cycle, and M1 is also further divided into, you can say, like prophase, metaphase, or pro-metaphase, metaphase, anaphase A, anaphase B, telophase, cytokinesis, like further stages. But we are not going to talk about those stages, but we are just going to talk about how they are regulated, means how they are connected with each other, and how all the phases of cell cycle are regulated by with the help of protein complexes and biochemical pathways and reaction like signaling pathways and etc. Okay, like any other videos on YouTube, you will find they always talk about the CDKs, cyclins, CDK inhibitors, anaphase promoting complex, all those protein. And one thing that I want to understand is that, like, so suppose that there is a cell present in just G0 phase, okay, and suppose that there is a like signal to proliferate this cell, and the cell is present in G0 phase. Okay, so how the G0 phase is reverse, so how the G0 phase is going to reverse into G1, and from G1 to S, from S to G2, from G2 to M, M to G1, and M to G0, because there are, at the end of cell cycle, there are two possibilities. Either it can go again into G1 and again start the cell cycle, okay, or it can go to G0 phase and just stop that cell cycle, that the process that is going on. Okay, so this is the main thing that we are going to understand in this video. Okay, how the biochemical pathways of each phases are connected with each other. Okay, because this is a cycle means every step of that cycle is connected with each other in a cyclic pattern, okay, in a cyclic way. And this is the thing that we are going to understand in this video. Okay, so without wasting any time, we are going to just start this lecture. And one more thing that I want to advise you, that if you want to understand the cell cycle regulation, you should already know that what is the cell cycle, what are the phases, what are the main events that are taking place in those phases. Yes, these are the main things. And more things are like what are the CDKs, what are cyclins, and CKIs, CDK inhibitor, what is the anaphase promoting complex. Okay, so these are the very basic thing that you should know about them. Okay, and if you don't know about them, just read in your books, you can easily find it. Okay, so let's start our actual lecture now. Okay? Let me draw a diagram first. Okay, so this is our diagram. Okay, and this is our cell membrane. Here it is outside of cell, and this is totally inside of cell. Okay, what is going inside of this cell? So there are many chemical compounds that can trigger the activation of mitotic cell division, and those compounds are generally referred as mitosin. Okay, we all know that. So for example, as a mitosin, we are taking here a growth factor. Okay, for example, we are taking here growth factor. And one more thing you should notice here, that every mitosen has its own specific signaling pathway with the help of which it trigger the activation of whole cell cycle. Okay, so this growth factor also has its own specific signaling pathway. So let's understand that. So this is our growth factor, and growth factor will come and combined with its specific receptor. Every factor has its specific receptor. This is a funny quote. Okay, so this growth factor will come and combine with its specific receptor. Now a very special process will occur here, that another receptor will come and combine with that receptor which is directly attached to GF. Okay, and both receptor will dimerize, and the kinase domain that are present in the intracellular part of that receptor will phosphorylate each other. Okay, and this trigger its activation. They will phosphorylate. There are kinase domains in the intracellular part of receptor that phosphorylate the tyrosine residues. Okay, now the tyrosine residues are phosphorylated. Okay, so there is a very specific protein complex named GRB2 and SOS. This is a very specific protein complex that will come and bind with those tyrosine residues that are already phosphorylated. Okay, so this protein complex will come and bind with that phosphorylated tyrosine residues. This is our protein complex, and SOS stand for son of sevenless. SOS is a family of guanosine transfer factor that works on Ras protein. Okay, so SOS has an activity to hydrolyze the transfer of GDP from GTP. Okay, GDP from GTP. Okay, SOS will hydrolyze the transfer of GDP from GTP. Okay, and this process also can be referred as Ras activation. Okay, Ras activation. Now the first step is now our complete. Now the activation of Ras protein will trigger a very specific kind of signaling pathway, and that pathway is Ras protein will trigger the activation of RAF, and RAF will trigger the activation of MEK protein, and MEK protein will trigger the activation of ERK protein. Okay, these all are kinases, means it can phosphorylate a very specific proteins. So ERK stand for extracellular signal regulated kinase, and MEK stand for something like MEK kinase. So now this ERK will phosphorylate some specific proteins that trigger the transcription of AP1 factor. ERK phosphorylates some specific kind of transcription factor or protein that will trigger the transcription of AP1 factor, and those factor AP1 stand for activator proteins. Okay, and here those factors are GUN Jun and FOS FOS. Now our signaling pathway is now completed, and we are going to see that how this signaling pathway will trigger the onset of G1 phase. Okay, let's see this. So in the DNA or in the genome of cell, there is a gene that have the coding language for cyclin D protein. Okay, suppose this is the gene, and this gene has its own promoter region. This is its promoter region, and this is whole coding sequence. And at the end, in this promoter region, there is a very specific site called AP1 site. So these AP1 factor will come and bind at AP1 site, and binding of those specific AP1 factor will trigger the transcription of cyclin D gene, and as a result we have our cyclin D protein, that is a regulatory subunit of CDK complex. Okay, so now we have seen that how the signaling pathway or how the binding of mitosin induce the activation of or onset of G1 phase through a signaling pathway. Okay, and every mitosin has its own signaling pathway, okay. So now we have our cyclin D gene. So let me explain some cyclins and how they are master regulators of all those phases of cell cycle. Okay, so let me explain something here. So let me write it here. For early G1, the master regulator are CDK4 6 cyclin D complex. Okay, they both are separate. CDK4 is combined with cyclin D and CDK6 cyclin D. These two complex are the master regulator of our early G1 phase. And for late G1 phase, there is another complex called CDK1 cyclin E. This is the complex that regulate the late G1 phase. And for our S phase and as well as G2 phase, they both are controlled with the help of CDK2 cyclin A complex. Okay, and for our M phase, there is a complex that regulate approximately whole M phase is our CDK1 cyclin B complex. Okay, these are our master regulators. Okay, so let us draw the diagram of the concentration level of those cyclin. It will be very helpful for you guys. Okay, so these are our phases like S phase, G2 phase, this is our M phase and G1 phase. G1 phase is the longest phase among all the phases of cell cycle. Okay, so if we see the concentration of level of our, for example, our these two, so cyclin D, CDK46 cyclin D complex, their level will be like something like this. So suppose that this is the early G1, so their level in early G1 will high and then 0. And if we see the diagram for this complex, so its level will be high in the late G1, and for CDK2 cyclin A, the level of this complex will high until the end of G2, and for this complex the level of something like this, because its level is always high in the M phase. So this is like the diagram pattern of the concentration of cyclins. Okay, this is our M and this is our S1. Here it is our late 4 and this is the okay. So this is just a very simple diagram to understand you in a better way. Okay, so let me erase this one. So now we have our cyclin D. Now this is active part. Okay, active cyclin D. So this active cyclin D will combine with the CDKs. Now remember that the concentration of CDK is literally just like no up and no down, just constant throughout the whole cell cycle. Okay, they are not going to degrade or any other process, metabolic process. They are always, the level of CDKs are always constant throughout the cell cycle. So now CDK is going to bind with its suitable CDKs like CDK4 cyclin D complex. Now we have this one, and CDK6 as cyclin D complex. Now we have these two complexes, and now we are going to see that how these two complexes regulate the whole G1 phase. This is our G1 phase is going on. So onset of G1 phase easily can be recognized by the synthesis of cyclin D. Okay, so now we have our these two master regulators, and we will see here that how they are going to regulate the G1 phase. Okay, so these two complex will phosphorylate retinoblastoma protein. Okay, so let's understand what was the necessity to do this and what can be the effect of the phosphorylation of RB protein. Okay, so there is a very specific complex called E2F RB complex. So E2F is a transcription factor that is necessary for the transcription of all those proteins, all those machinery that is necessary for the further cell cycle process or for the DNA replication process, like our pCNA, proliferation cell nuclear antigen, our DNA polymerases, okay, single stranding binding proteins, and all those proteins. Okay, so if we want to transcribe the genes of all those proteins, we have to activate the E2F transcription factor. And if the retinoblastoma protein is bind with the E2F transcription factor, means it is in its inactive phase. Okay, E2F if bind with RB protein, it means they are in inactive phase. Okay, so these two complex will phosphorylate retinoblastoma protein. So as a result, this retinoblastoma protein will dissociate from the E2F factor, and now E2F factor is our free, and now it is active and can transcribe easily all those protein, all those machinery that is required for DNA transcription. And to pass the restriction point, cell has to transcribe all those protein if it want to pass the restriction point. Okay, now the phosphorylation of RB protein trigger the activation of our E2F factor. Okay, and here we understand all those things, that how they are going to activate E2F. Okay, so phosphorylation of RB protein will trigger the activation of E2F transcription factor, and the activation of, because now E2F factor is free and it can easily transcribe all those machinery that is required for DNA replication. Okay, so this E2F will transcribe all that machinery like pCNA, DNA polymerases, single stranding binding proteins, and one more thing they are going to they are going to transcribe is that cyclin A, and as well as, okay, only cyclin A. These are some very important protein that are going to transcribe, that are going to translate it after the activation of E2F, and this is the phase also known as the passing of restriction point. If these machinery is synthesized, means cell has crossed our restriction point. Now a very important thing that we have to notice here is that GF binding, means the binding of mitosen compound, is only necessary until the cell has passed our restriction point. If a cell has passed the restriction point, there is no longer the necessity that this GF has to bind here. If the cell has passed restriction point, it will definitely go further for the cell cycle. It is not going to stop until any kind of DNA damage is going on in the further cell cycle. Okay, so if the cell has passed restriction point, there is no longer necessity for the binding of mitosen. Cell has crossed the restriction point and synthesized all those machinery and proteins which is necessary for the further S phase. Okay, and one more thing you have to notice here, that cyclin A is also already transcribed as a result of activation of E2F factor. Okay, so now after the synthesis of cyclin A, there is a very specific protein called CKI, means CDK inactivation or CDK inhibitors. They can inhibit the activation of cyclins. Okay, there is a very specific CKI named P27 that bind with cyclin A and inhibit its activation immediately after its synthesis. Okay, so there is a polyubiquitin ligase complex named SCF. And SCF is a protein complex made up of three proteins, SKP2, Chiolin, and F-box protein. We are not going to that deep level, okay, we have to just understand all those main steps. So now SCF will trigger the destruction of P27 inhibitor, and the destruction of P27 ultimately trigger the activation of cyclin A. So now we have our active cyclin A. Now remember, we have talked about only cyclin D, which is present in early G1. The only most important thing is the phosphorylation of retinoblastoma protein. In early G1, it is done with the help of cyclin D, and in late G1, it is done with the help of cyclin E. Okay, this is our main difference, and this is our whole G1 phase. And there is a one more very important process. We can generally refer to the assembly of pre-replication complex. Okay, the assembly of pre-replication complex is also takes place in our G1 phase. From the synthesis of cyclin D to the synthesis of all those machinery is our whole G1 phase. Okay, there is a one more very important phenomena is going on in G1, which is the assembly of pre-replication complex, and let's see how is this pre-replication complex is going to happen. We are just taking the example of one gene, that how the pre-replication complex is going to assemble in that gene. Okay, so there is a specific region called origin of replication, ORI region, and for the assembly of pre-replication complex, in first step very specific protein called CDC6 and CDT1 are going to assembled at the ORI region. Okay, this is our ORI region, and these are our both proteins CDC6 and CDT1. Okay, and in second step MCM, that is a helicase protein, are going to assembled at this ORI region. Okay, so this is our ORI region. Here is our CDC6, here is our CDT1, and there is our MCM proteins. Okay, and this whole complex are generally referred as pre-replication complex. Okay, this is the pre-replication complex, and the formation of pre-replication complex also takes place in the G1 phase. Okay, so there is a very specific question that how you are going to inhibit the re-replication of that same gene. Means what will cell do to prevent the replication of the same gene another time? So these kinase phosphorylate these two proteins CDC6 and CDT1. If they are going to phosphorylate, they are not going to assembled again on that ORI region, and this prevent the re-replication of that gene. Okay, that's the very simple thing. So now in G1 the pre-replication complex has assembled. Okay, and now we are going to understand that how the phases or events of S phase are going to triggered. So till now we have understand that how the binding of mitosin induce the activation of cell cycle phase with the help of a specific signaling pathway. Okay, and another thing that how the G1 phase trigger the onset of S phase, because this is our whole G1 phase. Okay, in early G1 cyclin D is necessary, and in late G1 cyclin E is necessary for the phosphorylation of RB protein, and this is our whole G1 phase till here, till this step. Okay, and the assembly of pre-replication complex is also going on in the G1 phase, already in the G1 phase. Okay, so now we have understand that how the G1 and S phase is connected with each other, how the events of every phase is connected with each other. So we have just understand the G1 phase, and now we also see them. So see, if we have the active cyclin A, it will combine with its suitable cyclin D kinase, which is CDK2, and it will combine with CDK2 and made the CDK2 cyclin A complex, and this complex will trigger our whole S phase. Okay, so now we have our this active complex CDK2 and cyclin A. Okay, so let's see how this complex will trigger the activation of S phase. Okay, so S phase, the main event that takes place in S phase is the duplication of DNA, duplication of the genome, and the synthesis of all the proteins that are necessary for the whole cell cycle. Okay, so we already have our pre-replication complex assembled. Okay, so now we have to activate this complex so that the whole DNA can be easily replicated, duplicated. Okay, there are also two proteins named CDC7P and CBF4P that help in the activation of pre-replication complex. Okay, so these two protein and this whole complex will phosphorylate our MCM proteins. MCM are the helicase, we all know that, and helicase are the protein that initiate DNA replication. Okay, so these two proteins and one complex will trigger the phosphorylation of MCM, means its activation, and then the DNA replication will take place. Okay, so after the completion of DNA duplication, now we have our DNA at the double amount, 2x DNA. Now means now we have our DNA as a double amount. Okay, and we also have all the proteins that are necessary for the further cell cycle. Okay, as in the S phase, all the DNA is duplicated and as well as the proteins are going to synthesize. Okay, so for example, what kind of protein? Okay, so now I'm going to erase this one because now we already understand how the pre-replication complex is going to assemble and how the activation of pre-replication complex takes place in the S phase. Okay, in S phase, this is this is a very easy step in the S phase, CDK2A. Just we have understand how this complex will trigger all the DNA duplication. Okay, and also the protein synthesis going on. So what kind of protein is going to synthesize here? So now listen, I'm explaining this thing in a very short and very simplified way. Okay, because when you read all those reference book, when I will tell you, okay, at the end of video, or you can simply check out the description box, they're a very highly described way, but I'm just explaining it in a very simplified way. Okay, so what kind of protein are there? For example, our cyclin B are there. Okay, APC complex are there, CDC20 is here, CDH1 is here. Okay, MYT1 is here, V1 is here. I have just picked up the proteins that are necessary for the regulation. Okay, but here all those proteins are available that are necessary for the further cell cycle. So the proteins, for example, cyclin B, MYT1, and anaphase promoting complex, or cyclosome V1, CDC20, CDH1, all those proteins. Okay, now we have all those protein in our S phase. And one more thing, here is a checkpoint. Yeah, in the S phase there is a checkpoint called Intras Checkpoint. Okay, let us understand what the checkpoint do. So suppose that during the DNA replication, any kind of DNA damage take place, any kind of DNA damage occurs by any mistake. Okay, so how that DNA damage is going to detect? So there can be of three type of main DNA damage. First is our double strand break, and second is single strand break, and third one is replication stop. Means the replicatory complex is just stopped in one place. Okay, these are the three major DNA damage that can occur. Okay, so now let us see that how those DNA damage are going to detect. So for example, here is our replication stopped. Okay, here is our single stranded DNA damage, here is our double stranded DNA damage. Okay, double stranded, single stranded DNA damage, replication stop. So when double stranded DNA damage takes place, it triggers the activation of ATM, and ATM stand for ataxia telangiectasia mutated. Okay, let me write here, ataxia telangiectasia mutated protein. Okay, this is the full form. So double-stranded DNA damage will be sensed by mRNA complex that is attached in that position where the DNA damage has taken place. So the double-stranded DNA damage will be sensed by our mRNA complex, and this will trigger the activation of ATM, that is stand for ataxia telangiectasia mutated protein, and this ATM will trigger the activation of our CHK2 protein, which is responsible for the stabilization of P53. Okay, P53. So P53 is a very important protein and also known as guardian of the genome, because it helps in the prevention of whole that dangerous mutation. Okay, and there are two ways. If the DNA damage is repairable, then P53 will trigger the P21 activation or P21 transcription, and that P21 will trigger the inactivation of CDK2. If CDK2 is inactive, it is definitely going to be a cell cycle arrest, because CDK2 is responsible for whole the process that is going on in that time in the cell cycle. But if CDK2 is itself inactive, then this whole cell cycle is going to in a arrest period. Okay, this is the way that how DNA double-stranded DNA damage is going to detect, and it will trigger the cell cycle arrest. Okay, so let's understand now how the single-stranded DNA damage is going to be detected. Okay, so single-stranded DNA damage and replication stop is going to detect with the help of ATR. ATR stand for ataxia telangiectasia and red9 mutated protein. Okay, red9 protein. Okay, not mutated, red9 protein. Okay, so this ATR will trigger the activation of CHK1 protein, and the CHK1 protein is responsible for the inactivation of CDC25 phosphatase, and this is a very important phosphatase because these are the phosphatase that remove the inhibitory kinase attached with those protein which are necessary for the cell cycle, and if they are inactive, then these inhibitory phosphates are not going to detach with those protein, and cell will ultimately trigger the cell cycle arrest. Okay, so these are a very simplified way that how a cell cycle is just related with each other and how it is going on a very fluent way. Okay, so this is how double Intras checkpoint works. Okay, so let's see. So this is our S phase. Okay, so now let's see that how the S phase will trigger the onset of G2 phase or M phase. Okay, but if you ask me that why the G2 phase is necessary, why it is present with the cell cycle, so I don't know. But if I ask you a question that why water is necessary for us, there is only one answer, that water is necessary for us because we can't live without that, because we can't live without water. So this is the answer, that our cell can't function properly, can't divide properly if the G2 is absent. Okay, this is a very funny answer, but it may be because that's why the G2 phase is present from the evolutionary time to till now. Okay, so let's see that how the S phase will trigger the G2 or our M phase. So the synthesis of cyclin B is continuous during the S phase and even in the G2 phase. Okay, even at the G2 phase. So in the S phase and whole G2 phase, cyclin B is constantly going to synthesize. Now remember one thing, that if APC anaphase promoting complex is already synthesized at the S phase, why this complex is not going to function? Why this complex is not going to function here? Because a very specific inhibitor protein is also going to synthesize, named as EMI1, and this protein complex inhibit the APC anaphase promoting complex all the time from S phase to G2 phase, okay, until it comes the M phase. So in the S phase, and so in the whole S phase and G2 phase, EMI1, this is the inhibitory protein that prevent the anaphase promoting complex activation. And one more question here, that why these two proteins are not going to function, because they are also phosphorylated with some kind of inhibitory kinases. That's why they are not going to function in this phase, okay. They will properly function in the M phase when that inhibitory phosphate will remove. Okay, so now we have our cyclin B protein, but it is now inactive phase. Okay, so it is now inactive cyclin B protein we have here. Okay, but why this is inactive? Because MYT1 and V1, these are two proteins that are responsible for inactivation of cyclin B. Yeah, because inactivation of cyclin B is also necessary in the S phase and G2 phase, because if cyclin B is going to be activated in S phase or G2 phase, there is a clearly mutation that is wrong, and this is not going to happen. Okay, so these two proteins also synthesized at the time of synthesis of cyclin B, so they can prevent the activation of cyclin B. But at the onset of M phase, cyclin B will activate. Okay, so now we have cyclin B with the help of CDC phosphatases. So these are two inhibitory kinases that prevent the activation of cyclin B. But at the onset of N phase, due to with the help of CDC phosphatase, cyclin B are going to activate and the inhibitory phosphate will remove, and now we have our active cyclin B. Now we see here that how these all the pathways are very beautifully connected with each other, and I bet you that you have never seen this kind of explanation in any kind of videos on YouTube, because this is my way and I am just explaining on my way. Okay, so now we have our active cyclin B, means we are in now M phase, early M phase. Okay, so this active cyclin B will combine with its suitable CDKs, that are CDK1 cyclin B complex, and the formation of CDK1 cyclin B complex will take place. Now we have our master regulator of M phase, our early M phase. And now we are going to understand that how this complex will initiate whole M phase. Okay, we just started from that signaling, from the binding of our growth factor, and we are just walking on whiteboard, and just our signaling pathway G1 phase, S, G2, and now we are in our M phase, early M phase. Okay, so we have now our this master regulator, and what this is going to do is the phosphorylation of CDC20 will trigger its activation. Now we have our active CDC20. Okay, now CDC20 is active and it can easily bind with anaphase promoting complex. Now remember that anaphase promoting complex can't function without the help of these substrate protein, because this complex itself even don't know that what kind of protein it will trigger. Okay, so that's why these substrate protein, these recognition proteins are necessary. Now CDC20 is now bind with anaphase promoting complex, and this will trigger the destruction of so see, this complex is not going to destroy proteins. It will just add ubiquitin to that protein, and that protein will be lysed with the help of proteasome. Okay, this is the concept. So this complex will trigger the destruction of very specific protein. So first is our cyclin B, and another is our securin. And one more thing that we should notice here, that this double amount of DNA will be condensed and take a form of chromosome. Okay, chromosome. Now these chromosomes, so suppose that these are those chromosomes, and they are just bind with, and these two chromatids are just bind with the help of very specific protein that are known as SCC1. Okay, this is the protein that help in the binding of both the chromatid. Okay, so destruction of securin. Now remember one more thing, that there is an enzyme called separase that will cut all those protein subunit that are helping in the attachment of chromatid. Okay, but this separase, if securin is bind with the separase enzyme, separase cannot work. That's why the destruction of securin will free our separase enzyme, and separase will cut all those SCC subunit that help in the chromatid binding. Okay, so this is the way that how the destruction of securin protein will trigger the chromatid separation in the anaphase. Okay, so we are directly writing here that destruction of securin will trigger the chromatid separation. Okay, chromatid separation. Okay, so when all the chromosomes are just separated and they are present at their poles, and the chromatid separation, when all the chromatid are present in their poles, it will trigger the activation of two protein. The first is CDH1 and another one is CDC14. Now we are at the end of M phase, means telophase, late telophase. We are here at the telophase. Okay, late anaphase or telophase or late M phase. So CDH1 is now active due to the whole chromatid separation. All the chromatid are just separated. It will trigger the activation of CDH1 and it will combine with anaphase promoting complex. Okay, so now this complex will trigger the destruction of our CDC20 protein, all those cyclins that are left behind in the cell cycle. Okay, so this is our late G1 phase, because everything is destroyed and now all the chromatid are just separated, they are reached to the poles. I have done a mistake that cyclin B is not going to be destroyed by APC anaphase promoting complex CDC20. This is not going to destroy by that complex, but CDC, listen to me here carefully. CDC14 will trigger the activation of SISIC1. Okay, so CDC14 will trigger the transcription of very specific proteins, SIC1 protein, SIC1 protein, and it will bind with cyclin B, cyclins that will inhibit the that will clearly inhibit the function of all the cyclin B, and they are now inactive phase. Okay, so cyclin B are just inactive, all the CDC20 are going to and now CDC20 is destroyed, all the cyclins are destroyed. And one more thing, that cyclin B destruction will trigger with the help of this complex and they are also destroyed. And now we are at the now our whole M phase is just completed in a short way. Now there are two ways. This was our late M1 phase, late telophase. Okay, all those just chromatid had just separated to their and reached their poles. Okay, and just cytokinesis is going to be happen at the late M phase. Okay, late M phase, there are two ways that cell will progress toward the G1 phase. Another time it will again reverse that process and this cycle is going on. And another possibility is it can go to the G0 phase and it will not trigger again that cycle. It will not going to initiate again that cycle. Okay, cycle again. So in the next video, because this video is going to be very long, so in the next video we are going to understand that how these two possibilities will take place, that how the late M phase will trigger the onset of G1 phase, and how the late M phase will trigger the onset of G0 phase. What kind of biochemical changes will take place in that cell to initiate these phases again. Okay, so this was the very simplified overview of cell cycle, and the main the main center of this lecture was that how these master regulators are going to regulate that phase, are going to initiate that phase, how all the biochemical pathways of cells are regulated with each other. So the main point of this lecture was that how these master regulator regulate just their own phase, and how all the biochemical pathways of whole cell is just related with each other. So this was my main point that I have to explain you. So overall this was the lecture. So please, if you enjoy this lecture, hit the like button, and if you want that, then subscribe my channel. Okay, then so in the next video we will talk about this concept. Okay, this one. So see you guys in the next video. Bye.